Hello, this is Alana,
And Macy.
and today we're going to be giving you an overview of our lab experiments this semester. This is Gardens of the Galaxy. Our research this semester was important because studying plants in space is key to understanding life sustainability during long-term space missions. Research focuses on how microgravity affects plant growth and development. Technologies like CRISPR enable genome editing, while nanopore sequencing allows rapid DNA analysis. Our model organism that we used was the Arabidopsis thaliana because it's easy to manage and it has a quick life cycle. Current experiments on this study are going on the International Space Station, where they grow plants like lettuce, cabbage, kale, and zinnias in microgravity to explore plant adaptation in space.
So, CRISPR is the Clustered Regulatory Interspace Short Palindromic Repeats. It is essentially a tool that edits DNA by acting like molecular scissors. So, it's a guide molecule that directs Cas9 to a specific DNA spot where it makes a precise cut, which is shown in the image above. We capitalize on the cell's repair process to disable a gene or add new DNA, which is a non homologous end-joining process.
The goals and objectives of this experiment this semester was to optimize plant survival to sustain human life in space and identify key genes that enhance plant adaptation to space conditions, comparing gene expression in tissues grown on Earth versus in space. Using the new CRISPR technology, our study aims to eliminate specific genes to improve plant growth and adaptability in microgravity. Another focus is to understand changes in root development under microgravity, identifying genetic factors that promote root growth and plant stress response. Additionally, nanopore sequencing will be used to analyze epigenetic changes between spinning and non-spinning plants, allowing for real-time monitoring of genomic modifications to better understand plant adaptation to space environments. In the beginning stages of lab, we started with gene bioinformatics, which is using various websites such as Thaliana and Blast to research the genome of the Arabidopsis thaliana. The second step that we completed was to use the CRISPR app within BenchLink to create our 20 nucleotide long sequence, which is the guide, which will be pictured along with the plasmid in the results section.
Okay, so this is our simplified version of the plasmid extraction process. Dr. O'Connor ordered the CRISPR plasmid, which was housed in E. coli cells needing to be extracted and isolated. So we used the QIAGEN mini prep kit protocol to do the isolation process. So BSA1 was added, and that is used for decreasing the enzyme loss on tube and pipette tip surfaces, and it also stabilizes enzymes in the reaction. So once that was added, the plasmid was then leaving the open cell, and it was centrifuged, and then that is what led to the plasmid being separated completely. After this process was completed, we started the gel electrophoresis, where we loaded samples into the wells with a green loading dye added to weigh it down, allowing it to settle to the bottom of the wells. We then applied a current, which DNA holds a slight negative charge that allows it to migrate to the positively charged anode. Once that was completed, we analyzed the gel bands with the UV light on.
After getting the results from the gel electrophoresis, we extracted the top band from the gel using a size 10 scalpel blade, and then we weighed that chunk of agarose gel, which was 0.57 grams. We then added 3 times the volume in ADB buffer, and then we incubated it at 55 degrees Celsius for 10 minutes. After it was melted, it was put into a spin column and then spun down with the centrifuge, and then went through multiple processes of washing, and the result is purified DNA. Using this DNA, we then ran it through a MinION sequencer to run the nanopore sequencing, and the results were relayed back to us via data on the computer.
Okay, so the process of nanopore sequencing, it involves reading the code of single DNA strands as they are threaded through extremely tiny pores, which are also known as nanopores, which is shown in the image. They are embedded within a membrane, and as the DNA moves through the pore, it creates signals that can be converted to read each base during this process.
As a result of our bioinformatics research, we found a gene that is significantly changed from growing on space versus Earth, and the fold change is positive, which means it's highly expressed in microgravity conditions. As you can see in Table 1, this is our gene ID data. We chose the gene AT5G27400 because it is significant in both the root and leaf tissue, and it has a positive fold change in both as well. We know that these p-values are significant because they are less than 0.05 for the root and leaf, and this is shown in Figure 1 in our volcano plot, where both dots for the root and leaf are above the red significance line. And both tissues are also in between the threshold of negative 0.8 to positive 0.8, meaning it's a reasonable fold change. Next, Figure 2 shows us the tissues of the Arabidopsis plant to where our gene is most highly active. Therefore, in the seeds during germination and the 9 to 11 budding stage, and the common denominator between the two, it is the production of seeds or the seed itself. This shows how our gene can be very important with the production of plants in space. For example, astronauts may need to produce plants very quickly to feed themselves, and by knocking out our gene, it could possibly help that mass production. Our next bioinformatics result is the phylogeny tree we got from the database of the Arabidopsis genome, and this shows us other organisms where our gene is found in, and ours mainly only shows up in plants, so this means it's specific to plants and their processes. An important thing to note in Figure 4 is that in the entire Arabidopsis genome, there was only 16 genes that were significantly expressed and had substantial fold changes. Although our gene was significant in both the root and leaf tissue, it did not have a substantial fold change on either, which would be a value of greater than 2 or more. As previously mentioned, Figure 5 shows the plasmid that was created by benchling, and from there we chose an early exon sequence to select our guide sequence, which involves 20 bases. Because it happened so early in the exon, the hope is that when Cas9 goes and cuts out this entire sequence, that it will change up the entire amino acid sequence thereafter drastically or completely knock out the gene. Our guide sequence is highlighted in blue in Figure 6, and it's 20 bases long so that the Cas9 cut is very specific and only cuts out that sequence.
Okay, so our next section is the nanopore sequencing results. It shows the read length graph that the total numbers of bases versus the read length. The longest 1% of strands are classified as outliers and excluded to allow focus on the main body of data. So our Figure 7 data shows that the read length that we produced was 679.936 bases. So for Figures 8 and 9, this is the methylation and microgravity resulting from our nanopore sequencing. The red marks on the image indicates that there is definitely methylation, and blue means there is a low chance of methylation. In Figure 9, you can see that there is an addition of blue and pink, as well as a change from red to blue, which was pretty interesting.
After completing the nanopore sequencing, we were able to find the number of mitochondria within the control, which was the ground cells, versus the cells of the plants in space. So we did this by using the FASTQ files given to us, and we would use the formulas below, which were genome coverage equals the total aligned bases divided by the genome size. And once we had the value for the genome coverage, we could use that in the second formula, which was the mitochondrial genome coverage divided by chromosome 5 genome coverage, and then that all multiplied by 2, and the result was the mitochondria in the microgravity cells was 32.31, and in the ground cells it was 24.5. Alright, based on these results, there is more mitochondria in plants grown under microgravity conditions versus on Earth. This indicates that more energy is needed to maintain growth in microgravity environments. Stress response also plays a role in this, which could increase the energy demands of the plant cells, causing there to be more mitochondria. Our gene is significantly expressed in both root and leaf tissue. and these findings are going to be used to further experiment with CRISPR-Cas9. Our results will be used to complete the next step, which would be to insert our CRISPR guide into the plasmid. Once it's inserted, the Cas9 targets the sequence in the genome. This will cause a double-stranded break, and this will activate the non-homologous end joining process specifically, and this is error-prone, and we rely on this. This will create a loss of function or gene knockout. After CRISPR has taken place, then the mutated seeds will be produced and then can be grown. And then that's when we can observe our gene of interest's impact on growth in microgravity plants versus Earth. This will determine if our gene is important enough to eliminate for the benefit of astronauts in long-term spaceflight. And lastly, we sincerely thank Kentucky Wesleyan College and the Science Department for providing the equipment and materials for our experiments. Special thanks to Dr. O'Connor for enhancing our understanding of genetics and laboratory safety, making this a valuable learning experience. And our references are provided below. Thanks for listening.